So carrying from where we left, the factors promoting the interaction between animals and people and potentially leading to the occurrence of Nipah virus include globalization, and with it the exponential human and livestock population growth, changes in economic development and land use, which are promoting the emergence of pathogens through activities such as the intensification of farming and forest encroachment. And obviously, these changes in economic development also increase the rate of contact between humans and animals, allowing the spillover of the pathogen into a new host. High densities of that host species facilitate pathogen establishment and transmission within the new host. In the case of the Nipah virus, well, this one spilled over from wild fruit bats to domestic pigs in Malaysia. The high densities of pigs in local farms appear to have facilitated the establishment of pig-to-pig transmission, and the pathogen spill over from pigs to humans. So basically, high densities of domesticated species are always associated with low biodiversity. The third factor is changes in ecosystems. So after the invasion from the pathogen, biodiversity may act as a source pool, allowing pathogens to jump from vertebrate animals to humans. If there is low biodiversity, then there is a high density of domesticated animals, which increases exposure to pathogens, just like I mentioned before. The third disease is Lyme borreliosis. So it's a vector-borne disease that's also termed erythema migrans and oligoarthritis. It was only recognized in the United States in the 1960s and 1970s. It is caused by a spirochete named Borrelia burgdorferi, identified in 1982. It's transmitted by ticks, members of the Ixodes family, and the natural reservoirs are deer and the white-footed mouse, Peromyscus leucopus. Since the 1980s, cases of Lyme disease have increased in the USA, so it's become like a case of public concern in the country. So here we can see the distribution of tick vectors. Basically, the affected population is found in North America, Europe, and Asia. Tick vectors are different depending on the region. So, for example, in North America, vectors are Ixodes scapularis and Ixodes pacificus. While in Europe and Asia, the vectors are Ixodes ricinus and Ixodes persulcatus. So, how is the disease transmitted? Well, basically, there's an infected nymphal tick, which feeds on small animals, in this case was the white-footed mouse, transmitting the pathogen. The mouse becomes infected but remains asymptomatic, so it serves as a continuous source for infection. Transmission to humans usually occurs by ticks in the nymphal stage because there's like an increase in outdoor activity which coincides with the nymph feeding cycle. The nymphs then molts into adult ticks, which then feed and reproduce on larger animals, in this case the deers. Adult ticks are transmitted by the deer to leafy areas where they lay their eggs. The eggs then hatch into new and infected larvae that acquire the Borrelia burgdorferi agent when they feed on infected mouse. The thing is that mouse can maintain the Borrelia burgdorferi, so they continually act as reservoir hosts. So basically here we can see animals acting as both natural hosts, in the case of the vertebrate animals, and vectors, in the case of the vertebrate individuals. So how can we see here human involvement in the dissemination of Lyme disease? Well, basically through ecological changes due to agricultural or economic development. So basically, these changes favor an increased population of the pathogen or the natural host. Ecological changes can either be reforestation. So in this case, reforestation leads to an increase in the population of deer and the deer tick, which is the vector of Lyme disease. And the migration of people into forest areas. So this brings them in close proximity to the vector. And loss of biodiversity related to the host species, well, if the amplifying host species persists, then the disease risk will consistently increase as well if biodiversity declines. So this leads to an increase in the pathogen transmission. In this case, for this disease, the white-footed mouse is simultaneously the most abundant, competent, and highest quality host. Therefore, if biodiversity is lost, the host remains and it has a strong amplifying effect. And since it's the vector of the disease, well, the disease will persist. The other factors are climate change. Usually climate change helps determine tick populations and affect the spread of the disease. Basically, by providing conditions that are ideal for tick survival, and these include temperature and humid zones. For these ticks in particular, temperatures over 7 degrees Celsius and humidity conditions of 85% are ideal for their survival. Global climate changes, well, when there's an increase in temperature, this obviously also generates changes in tick population by either speeding and extending the vector's developmental cycle, increasing egg production, or increasing tick population density. After all, these are the temperatures that are most ideal for the tick to actually function. So season, basically this one is related to the tick's development or feeding cycle. We'll see a graph later on, but for example, in late summer is when the larva hatches and feeds on the blood of small animals, so in this case the white-footed mouse. Usually in winter they hibernate and their activity ceases almost completely, at least until late spring. Following spring and early summer, the larvae are transformed into nymphs and obviously start feeding again. And then during the fall, the nymphs transform into adults, and obviously they will start feeding on large animals, in this case the deers. So here we can see the graph, how it evolves depending on the season and depending on the month, and it has related to the development cycle of the ticks. The other factor related to climate change is rainfall. So there is a relationship between the amount of rainfall of precipitation and the incidence of Lyme disease. So usually it has to do with the loss of water regulatory controls of the tick. So what happens is that after the nymph ticks feeds on and infects their host with Lyme disease, they lose their ability to effectively control their water content. So if there is little rainfall, then many ticks may die following feeding because of this loss of water regulatory control. If they die, well, they don't reach adulthood and they don't lay eggs. 2 years later, the offspring population is reduced and thus Lyme disease incidence decreases as well. It also has to do with the control of white-footed mouse population. So if there's a decreased rainfall in the summer, it decreases the mouse food source, which is mainly vegetation. And this vegetation is what they need for winter. This leads to a decrease in the population of the tick's primary host. Decreasing the population of the tick's primary host will reduce tick populations and Lyme disease incidence as well. Bovine spongiform encephalopathy. It was first diagnosed in cattle in the United Kingdom in 1986. The etiological agent is a prion found in meat and bone meal used for cattle feed. The disease has so far occurred in Europe, Asia, the Middle East, and North America. It's basically a disease that groups several diseases into what is known as transmissible spongiform encephalopathy. So within this group, there's scraping sheep, chronic wasting disease in deers and elk, Creutzfeldt-Jakob disease in humans, and obviously bovine spongiform encephalopathy. It has a very long incubation period of 4 to 5 years. And it's a public health risk, basically in the form of the Creutzfeldt-Jakob disease, because it's a variant or bovine spongiform encephalopathy. It contains the same agent. Actually, since 1996, there has been 45 human cases that have been recorded, obviously mainly in the UK. So here we can see the geographic distribution. So mainly we've seen them in cases in Europe and in North America. So for example, the UK has reported 180,000 cases between 1987 and 2007. The United States, well, the first case of bovine spongiform encephalopathy was reported in 2003, which was the same for Canada. The first case was also reported in that same year. And since 1987, 25 countries in North America, Europe, and Asia have reported cases of this disease. So, how is the transmission? How does it take place? Well, basically, the cow eats food that's supplemented with either sheep bone meal, and this contains infectious proteins which are known prions. Obviously, since then, there has been a ban that has just prohibited this kind of feeding since 1997. Usually the prions, what they do is they curb normal protein production. They are absorbed in the stomach, so once they go inside the organisms, they are absorbed in the stomach, and then travel along the nerve fibers to the brain stem, and they just completely destroy the brain tissue. Obviously, they can also infect humans. So when humans eat all the processed meat products that might quite possibly contain prion, well, they obviously can end with a similar fatal syndrome. But this one is dubbed as a variant of Creutzfeldt-Jakob disease. So what happens is that the person eats the contaminated food, right? Generally, the prions are found primarily on the brain and the spinal cord of the infected animal. So after the person ingests it, the prions will travel to the brain, and they can remain dormant for a year. Then, when a disease attacks the nervous systems, the outer layers of the brain develops tiny holes, so it kind of acquires a spongy-looking appearance, and the person develops seizures, and eventually death may occur. So the interaction between livestock and people that led to the occurrence of bovine spongiform encephalopathy included industrialization in food production, particularly involving products of biological origin. So, as mentioned at the beginning of my presentation, modern and efficient operations need reduced costs, but there's also higher risk of accidental contamination with amplification of said effects. Which was actually the case of bovine spongiform encephalopathy in Britain. It resulted from interspecies transfer of scrapie from sheep to cattle due to an incomplete inactivation of the agent in sheep by products that were later fed to cattle. There's also, the fact that nowadays national and international animal transport / trade takes place. This contributes to the introduction of a potentially pathogenic agent into naive species, population, and environment. So there's just risk of exposure. And polyparasitism, because agents will come in groups. They won't come just by themselves. And this usually happens when they cross borders. So they obviously pose a serious risk in the emergence and spread of infectious diseases. And obviously overpopulation, because it increases the demand of food, both animal and plant source. Well, conclusions. Most emerging diseases are caused by pathogens already present in the environment. Changes in demographic, geographic, and ecological conditions may aid them in infecting new host populations, either animal or human. Other emerging diseases can be a product of a new variant, which is a rare case, but these variants or strains may evolve and cause a new disease. Anthropogenic and natural causes are responsible for changes experienced within the economic development and leisure area, human demographics and behavior field, travel and trade system due to globalization, and biological environmental area. Animals perform a very important role in zoonotic diseases, be it as a natural host or as a vector. They are also affected by an environmental, agricultural, demographic, behavioral, cultural, and socioeconomic drivers of pathogen emergence and disease incidence. And in order to develop preventive measures for combating disease outbreaks, it's important to carry out a rapid surveillance program. And that's about it. Well, thank you guys for listening, and I hope you enjoy the presentation. Thanks.